Good afternoon, everyone. My name is Katrina, and today I'll be presenting my Keystone project entitled Genetic Variant Analysis for Pathogenicity, Assessment and Implications for Disease Risk and Immune Modulation. Before I begin, I would like to thank my advisor, Dr. Andrew Taylor, for his guidance throughout my project. And in this research, I focused on understanding how small changes in the MC1R gene can affect a person's risk to certain diseases, specifically in regards to impact on skin pigmentation, as well as immune system regulation. Okay, introducing two main components of this research project. Firstly, looking at AMSH, which is short for alpha melanocyte stimulating hormone, is a peptide hormone involving important in several biological processes. Specifically, it regulates inflammation, controls skin pigmentation, and by influencing melanin production, as well as helping with energy balance. Additionally, the MC1R gene, or melanocortin-1 receptor, is the primary receptor for AMSH and binds to in the skin and in immune cells. So when, as you can see in the figure, when AMSH binds to MC1R in the cell membrane, then it triggers a cascade event in the cell, which then is crucial for skin protection as well as immune homeostasis. Under normal conditions, MC1R protects us from UV radiation by promoting the production of eumelanin, which is the darker pigment that absorbs and dissipates UV rays. However, genetic variants that reduce MC1R's function are associated with many traits which can include red hair, fair skin, as well as poor tanning ability, all factors which can be associated with higher melanoma risk. In addition to MC1R's role in melanoma and skin pigmentation, it also has roles in inflammation, which means that variants may impact and increase susceptibility to autoimmune and metabolic diseases. So the research question for this study was, how do genetic variants in MC1R influence pathogenic potential related to immune regulation, skin pigmentation, and disease susceptibility? Answering this can help identify individuals at higher risk for diseases linked to MC1R variants. So what did I do? So I started by looking specifically at the MC1R gene, and I specifically looked on chromosome 16, where the MC1R gene is located. I compiled 4 annotated variant files using a whole genome analysis from the 1,000 Genome Project, in which it has over 3,200 individuals. After cleaning the data and removing 241 duplicates, I had a final data set of 1,576 unique MC1R variants. After processing, all processing, visualization, and statistical analysis for pathogenicity predictions were conducted using RStudios, which is a programming language commonly used with genomic data. So now looking at the specific mutations, as you can see, the majority of mutations, specifically 1,438 of the variants, were non-coding in non-coding regions, which are areas of the DNA that don't directly impact the code for the protein. So these are typically not seen as of great interest when it comes to pathogenicity. Additionally, 59 were missense variants, where a single amino acid change could alter MC1R protein structure or function, and 5 were high impact, such as stop gain and frame shift mutations, which were likely lead to complete loss of normal protein function. So missense and high impact variants are of the greatest interest when it comes to pathogenicity. So specifically, to predict which variants were harmful, we conducted 3 computational tools. This was SIFT, PolyPen2, and CAD. SIFT, which is right there, predicts whether an amino acid substitution is likely to disrupt protein function based on evolutionary conservation. Polypen 2 evaluates the structural impact of amino acid changes, classifying them as either benign or damaging. And CAD integrates multiple annotations into a single score, and a CAD score greater than or equal to 20 suggests a strong likelihood that a variant is destructive. In total, 64 variants passed at least one of these filters, meaning that they were predicted to significantly impact MC1R function. Additionally, what we did is we identified 2 specific variants that stood out for having strong likelihood of being pathogenic. These 2 were RS 18007 and RS 1805008. These 2 variants had the highest CAD scores and are strongly associated with impaired MC1R signaling, reduced eumelanin production, and significantly increased melanoma risk using previous clinical studies. Their strong predictions impact in my analysis supports the evidence that they are high priority variants when it comes to clinical risk assessment. Additionally, we looked at disease potentially associated with MC1R, and unsurprisingly, melanoma was found to be the #1 disease associated with MC1R variant risk. But additionally, on top of that, we found associations with autoimmune diseases as well as diabetic retinopathy right there. This suggests that MC1R dysfunction has broader roles than just skin health, but also may impact the immune system regulation and chronic disease development. In conclusion, genetic variants in the MC1R gene can significantly alter disease risk, affecting both pigmentation-related conditions like melanoma, as well as immune-mediated diseases. My findings highlight the potential for early genetic screenings to identify individuals with higher risk and inform them about personalized medicine strategies, as well as additionally targeting AMSH, MC1R pathways could offer new therapeutic approaches for inflammatory and metabolic diseases. Thank you very much.
All right, we have time for a few questions. Yeah, in the back.
You mentioned melanoma and immune issues, immune diseases. Does this have any applications for immunotherapies for melanoma?
Yeah, that's actually something that is why I wanted to look into MC1R is I have an interest in immunotherapies, and AMSH, it's a relatively newly thing being looked into as potential, but it does offer immunosuppression specifically in the eye. So there's potential talks about it being utilized for immunotherapy development.
All right, any other questions? Yeah.
Thank you, great work. So you say early screening for individuals. How long does it take, I'm engineering but not bio area, how long does it take to go from studies like what you're talking about here, where you're identifying possible connections, to actually having it get out to being a screening tool that's used in the medical field?
Yeah, so there's currently rapid screenings already occurring. Genetic screenings are becoming more rapidly used. This research is to also highlight which areas of the genetic screenings to specifically look at for causing potential diseases, specifically individuals like in high risk. So the time frame is a little bit unknown, but genetic screenings are currently in development and they're increasing in use.
Great. Any other questions?
Did you find out anything surprising to you when you went through this process?
I would say the most surprising thing I found out was I didn't think I was going to be able to get results on my original interest, which was diabetic retinopathy, because it is a very understudied field, because it doesn't, there's not a lot of funding in that area and it's not looked at a lot. And specifically with AMSH, it's almost exclusively looking at melanoma. So I was very surprised that I was able to find clinical evidence of association with diabetic retinopathy because that took a lot of looking through data sets and not finding the clinical data that I needed to originally assess diabetic retinopathy.
Great. Any last questions? No? Awesome. Well, thank you so much.